Good morning, my name is Valeria Arroyo. I am going to talk about gene editing of CCR5 in autologous CD4 T cell of person infected with HIV. Definition: CCR5 is the main core sector of the human immunodeficiency virus, according to the studies that have been carried out. It has been demonstrated that the specific modification of the gene site, in this case, the infusion of autologous CD4 T cell in which DCCR5 gene was made permanently dysfunctional by a seen firmware nucleus X size.
First, let's start with the basics. This is a gene. Genes are made up of DNA, which are the blueprints to build the enzymes or proteins that do the work for your body. As far as we know, humans have between 20,000 and 25,000 genes. And most people have two copies of each gene inherited from each of our parents. They are the building blocks of our physical makeup, from the color of our hair, to our athletic abilities, to our immune system. But sometimes genes aren't built correctly. And a small adjustment to the DNA within our genes can change how our proteins work, which can affect how we breathe, walk, or digest food properly. To better understand how genes change, here's Dr. Michelle Kalos.
So our genes are mutating all the time as a result of chemical processes in the cell, as well as external influences like ultraviolet light. We can also inherit mutations from our parents. Now, when we wind up with these mutations, they can sometimes cause disease, and that's really where gene therapy comes in.
If genes are like the blueprint to our body, gene therapy can fill in some missing drawings. Gene therapy is the introduction, removal, or change in genetic material, specifically DNA or its molecule cousin, RNA, into the cells of a patient to repair or change a gene to treat a specific disease. The transferred genetic material then changes how a protein, or group of proteins, is produced by the cell. It can reduce levels of certain disease-causing proteins, increase production of disease-fighting proteins, or even produce new or modified proteins within the cell. You may be curious how a gene and the genetic material it holds is transferred to the cell during gene therapy. Well, a gene cannot be directly inserted into a cell. Instead, a carrier, also known as a vector, helps deliver the gene. Typically, viruses are used as vectors because they've evolved to be very good at sneaking into and infecting cells. But in this case, their motive is to insert the gene or genes into the cell. The infectious part of a virus is removed, allowing it to still enter the cell's nucleus without making the patient sick. It's worth noting there are two kinds of gene therapy, in vivo and ex vivo.
In vivo gene therapy, the genes are introduced directly into the patient's cells while the cells are still in the patient's body. In ex vivo gene therapy, cells are removed from the patient's body, then the genes are added to those cells in the lab, and the cells are then returned to the patient. The right approach depends on the disease that the patient has and the tissue that's affected, and that would tell us which is the best way to approach this.
Advances in gene therapy research allow us to treat and hopefully
Application in medicine. Investigations showed that 12 patients were considered in an open label, no randomized new control studies of a single dose of CF and modified autologous CD4 T cells. The patient had chronic arrhythmic HIV infection while receiving highly active antiretroviral therapy.
It's this incredibly tenacious virus that causes AIDS. And for years, we've come to think of it as something that can only be managed, but never cured. Managed thanks to the antiviral drugs we have today, drugs that made it possible to survive what was once a sure death sentence. But the drugs aren't a cure. They can keep the virus in check, but if you stop taking them, the virus will snap back. It will come back. And these drugs are still not available to millions of people around the world infected with HIV. But we now know from a few patients so far, a cure for HIV is possible. And that cure came from a bone marrow transplant. And that's something I'm very familiar with because I am an oncologist, a cancer doctor. And for more than 25 years, I've been treating cancer patients by giving them bone marrow transplants. And now I want to take what we've learned from these years of transplant experience and combine it with these new advances in gene therapy and gene editing and develop a cure for HIV that does not require a cancer treatment, a bone marrow transplant, but a cure that could be given through a simple injection, a shot in the arm, and made available to anyone living with HIV, even in places that lack medical infrastructure. So to understand how this came about, let me start with the Berlin patient, the first person ever cured of HIV.
Pros and cons. Pros. The primary outcome was safely as assessed by treatment-related adverse events. The blood level of HIV DNA decreased in most patients. Cons. Discontinuation of antiretroviral treatment 4 weeks after the infusion of 10 billion autologous CD4 T cells, of which between 11 and 28% were genetically modified with CFN.
A world without HIV. At American Gene Technologies, we think we're on the eve of just that. This year, we may prove that a functional cure is possible and see the end of HIV in our lifetimes. Gene and cell therapy is a new drug development modality that opens opportunities to get into the root causes of disease and even allows us to reprogram immune cells to be effective against HIV in a way that a natural body is unable to be effective. What we've developed is something called a functional cure. If this works, it could mean that HIV-infected individuals could go from lifelong antiretroviral therapy to basically living a normal life.
Our program is putting forth a highly innovative product that hasn't been seen before in the clinical world of HIV medicine. HIV is a unique virus for many reasons. One of the most important is that it attacks T cells. T cells are the drivers of your immune responses, and that means without T cells, you're unable to combat viral infections. So we have to make a change in those cells while we're working with them. And in our case, we put a very tiny amount of DNA into each cell, and that little bit of DNA programs that cell to resist HIV.
And so as a result, those T cells can do their job. They can clear HIV like you clear a cold. We can take HIV T cells and we can make them immune to HIV. Once they're back in your body, you have an immune system that's actually capable of fighting this otherwise very fragile virus. And we believe that's going to lead to a functional cure.
I started taking HIV meds 1 year after my diagnosis. I've been on just about every HIV medicine there is because once you take the HIV meds, you can develop resistance to them if they're not taken properly. But I would like to see the day where I don't have to take HIV meds anymore.
The traditional pharmaceutical industry has been highly successful at treating HIV, and as a result, they just haven't had the same incentive as biotechs to really go out there and swing for the fences and bring a cure. Well, AGT was founded to make a cure. The only way we're successful is if we can bring a functional cure to this audience. So that's what our focus is. That's where all of our incentives lie. That
Use implique in Ecuador. A serious adverse event was associated with infusion of CFN modified autologous CD4 T cell and was attributed to a transfusion reaction. The median CD4 T cell count was 1,517 per cubic millimeter at week 1, and significant increase from the pre-infusion count of 448 per cubic millimeter. In conclusion, CCR5 modified autologous CD4 T-cell infusion are safe within the limits of the study performed. Thank you.